Okay, so what the compartmentalization means is that, you see, we have some substances in the body which are produced and also they are being destroyed. And when this happens, there is no point for simultaneous occurrence of both. So when a product is being produced simultaneously, meaning that they occur at the same time. But for compartmentalization, it doesn't happen like that. When the substance is being produced, it is not being destroyed at the same time, but rather when a substance is produced, it takes some time, then it will be destroyed. You get it here. So generally, they are the synthetic. Synthetic means produced, and the type of pathway that helps in the production of substances is the anabolic pathway, and the breakdown, which is the catabolic. So these pathways, they are responsible for the synthesization and the degradation of what substances in the body. You get it? Yeah. For instance, enzymes for fatty acid synthesizes are found in the cytosol, whereas enzymes for fatty acid oxidation are present in the mitochondria. So what this is saying is that for enzymes which produce fatty acid, they are found in the cytosol, and but for enzyme which they degrade or oxidation means you degrading um the fatty acid. So these enzymes, they are also found in the what mitochondria. So you see that they don't occur at the same time. This will be produced in a certain organelle, then the other two will be produced in another what organelle. You get it? Yeah. So basically that's all about compartmentalization. So this will all occur depending on how the body needs it. So when the body needs fatty acid, it will be produced. When it doesn't need it, they will all be oxidized. So kindly take note of this table as well. We have the organelle, then the enzyme or metabolic pathway it is involved in. So let's move on to non-protein enzymes. So the non-protein enzymes are also called the riboenzymes. So some of the proteins, they are not necessarily made by proteins. Yeah, sorry, some of the enzymes, they are not necessarily made by proteins, but some are non-proteins. So these non-proteins are known as the riboenzymes, and these riboenzymes, they they have a ribonuclease P. It is very examinable. They have a ribonuclease P um in their system, and also these riboenzymes, they are being formed by a group of ribonucleic acids. Yeah, ribonucleic acids. So some scientists are also part of how, or they are part of how these riboenzymes were discovered, and some researches about it. Yeah. So we have the international units, the IU. So the IU is just some units for calculating for some of the enzyme activity that occurs. Yeah. And normally when you look at the unit of enzyme activities, um enzymes are never expressed in terms of their concentration, but rather expressed only as activity. So enzymes, they are not expressed as in terms of their concentration. So we don't have any units for enzyme where you get milligram and those stuffs. No. So enzyme, their unit is just expressed in what activities. So you can just see some examples being given: the Armstrong units, Somoza units, and so forth. Enzyme degradation So what this is saying that enzymes are not immortal. Enzymes are not immortal. That means enzyme they can die. Yes, they are not immortal because they can die. So there's a lot of variability in the half lives of individual enzymes. So each and every individual enzyme has its um half life, the period it can last for. Yes. So some are in minutes, hours, and stuff. So we have amylase being surviving for 3 to 5 hours, LDH being surviving from 5 to 6 days, and so forth. Enzyme synthesis. So for enzyme synthesis, we have some types of enzymes which are um, constitutive enzymes and inducible enzymes. Constitutive enzymes, they are housekeeping enzymes. So that means they are fairly constant in what they are doing and they are not controlled as well. Inducible enzymes, as the name goes, they can be increased or decreased per what the body needs. So let's move on to some applications of enzymes. Some application of enzymes. So for this one, you can just go through it. The applications are based on the categories. So we have therapeutic application, analytical application, application in genetics, industrial application, enzymes in disease diagnosis, enzyme in disease diagnosis again, then the slice end. So for example, what this is saying that in therapeutic application, so in therapeutic application, we have some enzymes such as the alpha antitrypsin, asparaginase, and urokinase. So therapeutic application meaning that these enzymes they help in the treatment of diseases. Do you get it? Yeah. Therapeutic means um healing or treatment of what diseases. So these enzymes they are involved in that action. So you can just glance and learn it. Normally, you can get some questions about it. Yeah, and analytical application. Analytical application. So for this one, meaning these enzyme like urease, cholesterol oxidase, uricase, lipase, luciferase, and the others. So these enzymes being analytically applied means it helps in studying our thinking abilities and some every research or some activities concerning thinking or understanding of something. So these um, enzymes they can be used for those actions. So normally you can have logical reasoning, problem solving actions and those stuffs. You can use these enzymes to help you in that. So application in genetics, we have industrial application. That means it is being helped in industries for genetically wise. These enzymes also helps in gene transfer and those stuffs. Okay, we have enzymes in disease diagnosis. That means they help in determining the types of diseases or types of conditions um, a body has or a body is suffering from. Yeah. So we are done for enzymes. Thank you all for participating. So what I will tell you is that just when you have time, go through the table. Sometimes we normally get questions on it, and you are good to go. Okay, thank you. Bye.